Good morning, everybody. It's my pleasure to be back in Croatia. I first came here 15 years ago with Ivica and his colleagues to take part in a series of workshops in Dubrovnik, and those still remind me of one of the best times of my international research career. Expert group in neurodevelopmental biology, and we've continued to collaborate with people from Croatia throughout this period. I have the graveyard shift. That means that I have to be quick to make sure that I don't keep you from your lunch. It's fortunate that people have been speaking ahead of me that will cover a lot of the territory that I will be able to skate over quickly. But I'll be talking about some of the international initiatives that do similar things to what you've heard about this morning, and how we brought some of these initiatives together within the context of HIBALL. So I'll start with a discussion of Healthy Brains, Healthy Lives, which is a large neuroscience research program at McGill. Its vision is to create a global center of excellence in neuroinformatics, and the application and the translation of those methods and tools to discovery science in brain health. The HBHL is split into 4 major themes: a cross-cutting methodological platform on neuroinformatics and computational modeling, and then 3 application domains in mechanisms of neurodegeneration, cognitive neuroscience of brain plasticity, and population neuroscience and brain health. We've hired at this point 19 new faculty, most of them are data scientists. Just to give you a flavor, the informatics and computational theme by itself has many different components, and these are all variations on a theme of network modeling, computational infrastructure that allows us to explore brain circuitry at the level of genes or systems neurobiology. We have the applications that I mentioned. Principal among these are connectionist models of propagation of misfolded proteins, for instance, beta amyloid in Alzheimer's disease, or tau protein in Alzheimer's, or indeed Parkinson's, and so on. And so all of these are propagation models which require big data analytics. Theme 3 is the applied cognitive neuroscience of brain plasticity, where we're studying sleep mechanisms, and again, connectionist pathways to underpin the neurobiology of sleep and normal cognitive processing. And theme 4 is a population neuroscience and brain health. And this is at 2 levels. It's looking at issues of stressed brain organization, nutritional deprivation, spousal abuse, alcohol abuse. All of these factors will affect the resilience of the brain. And we're also looking at psychiatric disorders within theme 4. Overall, the HBHL program has about 200 funded PIs and about 500 HQP, highly qualified people, and about 250 postdocs and graduate students. So as you can see, it's a large project. And over the last 5 years, we've produced about 1,000 publications at this point. Of course, all of this scientific industry requires a significant number of core platforms. These are all of the platforms that we use, but most salient to this meeting is the NeuroHub platform. And as you heard, we have to accommodate a wide heterogeneous community of users, from people on the right-hand side who really just want to upload and download data, to people on the left-hand side who want to take multimodal data. They want to apply advanced analytic workflows to these data. They want to use machine learning approaches. So there's a continuum of data handling burden and scalability that we have to address. Now, we take care of all of this within the context of HBHL with what we call NeuroHub. Brian Caron described this. And while there's a lot of similarities between what you heard from eBrains and what's going on here in terms of allowing us to collect the data, to process the data, to publish that data in an open format using tools such as as NeuroLibre, I will focus just briefly on the two major components of this, LORIS and CBrain. LORIS is the multimodal, multi-time point, multi-site databasing environment that allows us to capture imaging data, summary genetic and epigenetic information, behavioral clinical information, more laterally biospecimen information, blood, CSF, iPSCs, all of that kind of information, and electrophysiology, all of it all under one roof. All of that is then can be analyzed within the context of CBrain, which you heard from Brian, as a portal to the high-performance computing infrastructure originally of Canada, but then more laterally internationally, and if necessary, cloud services. So as I just said, this started life across the Canadian landscape of HPC sites, but now CBrain is a portal to, for instance, 3 of the Exceed sites in the USA. And we're developing linkages with sites such as Surfsera in the Netherlands, and we're working with Jan and the eBrains team within the context of the Jülich site in particular to develop interoperability between CBrain and Jülich as a prototype for what will eventually become a linkage between CBrain and eBrain in the European context. As Brian pointed out, CBrain is used in about 200 sites in about 30 countries around the world, so it's a well-established, mature infrastructure. Some of the applications that make use of this infrastructure, I'll make brief reference to. CBIG is an open biospecimen repository that anybody can get access to. This can be made available as either controlled or fully open access for biorepository, biospecimen information. I'll talk about a few of these projects as we go on. The CCNA is the Canadian Consortium on Neurodegeneration and Aging, and this is, if you like, a Canadian equivalent of ADNI, which I'm sure you're all familiar with. This is approximately the same size as ADNI, but it's interesting that the kinds of data that were collected within CCNA are quite broad in terms of their inclusion criteria. So they're not very, very restrictive in terms of just Alzheimer's disease. So this includes people with frontotemporal dementia, semantic dementia, vascular dementia. And at the time, I thought this was potentially a bad idea because it would be too heterogeneous. But now with the rise of machine learning approaches and advanced pattern classification approaches, I think this is actually a strength because this is closer to the real world. Most brain disorders are full of comorbidities, and we can use machine learning approaches to find the underpinning of each of these subclasses across this heterogeneous cohort. So this data set is available to people who want to explore it. We're making these data available through the Canadian Open Neuroscience Platform, CONP. You can go to this website. And CONP brings together sites across the country. It has an organizational overarching management environment, but it has developed the interoperable IT infrastructure, similar to NeuroHub that I've just described, that makes use of LORIS, CBrain, DataLad, all of these tools, to allow us to share data across the national and international communities. It has a training regimen where we have taken about 60 individual students and cross-trained them from, for instance, a genetics background to learn about imaging, or a psychology background to learn about mathematics and statistics. And of course, this is the essence of data neuroscience today, is multidisciplinarity. We've developed an ethics and data governance environment to allow us to understand the landscape of data sharing, both nationally and internationally across the various jurisdictions, which I'm sure that people in the Human Brain Project are more than familiar with, the issues of crossing national boundaries. At this point, this slide is actually somewhat out of date. This says that we have about 80 individual containerized workflows. The number is closer to 120 at this point, and about 60 different data sets of various flavors which are made available to the international community. Now, CONP provides a data sharing infrastructure to support numerous different platforms, and one of them that I'll make brief reference to is EEGNet, and this, as the name implies, is focused principally on electrophysiology. And it started life with the MNI Open Intracerebral EEG Atlas from Birgit Frauscher. And this is data that's derived from epilepsy patients where you're trying to find areas of the brain which are normal from depth, using data from depth electrodes. And as you can imagine, that is a delicate balancing act because you're looking at an epileptic patient to try and find areas where there's normal intracerebral EEG. So this is collected across hundreds of individual patients in 3 different sites internationally, and all of this was brought together in 2018 as an open intracerebral EEG atlas. Moving on to where we've taken this was part of the origins of EEGnet, and this is a national platform that is aimed at developing a unified platform for sharing and analysis of standardized EEG data sets to the national and international research community. And we want to develop a standardized way of sharing data. That means coming up with a common format, common analytics, something which requires a lot of work across a very heterogeneous international EEG community. When talking about the EEG international community, of course, we extended this beyond Canada to include what we call the Global Brain Consortium. And this is focused on EEG, behavior, interoperability, and outreach to low and middle income countries, because a fundamental driving force behind the creation of the Global Brain Consortium is that we want to be able to reach out to countries which do not necessarily have access to 7T MRI machines or advanced positron tomography systems. Everybody can get access to the inexpensive EEG technology. And so this GBC for the outset is focused on EEG-related applications. For the 2 reasons. It provides us with very high spatiotemporal dynamics in 4D, the data itself, and the fact that it can be applied in low and middle income countries. We've had numerous workshops over the intervening years since the launch, and the project GBC is organized into 6 projects on child development, malnutrition, COVID, Parkinson's disease, dementia, and one methodological project on global EEG norms. And I'll just focus on that for a second because this is more oriented towards a methodological and tool workshop that we have here today. So the Global EEG Norms Project, led by Pedro Valdez Sosa and Jorge Bosch-Bayar at our lab in Montreal, it's published its first usable tool is the dissemination of a harmonized quantitative EEG analytics environment and toolbox, and this is made available, deployed on CBrain. We've also developed an EEG to BIDS wizard tool, allows us to anonymize and standardize the data sharing of electrophysiology data across 9 different countries that have taken part in this initial international harmonization project. We've also added in work from Robin, Scott McCaig, Truong, and Delorme to allow us to develop a hierarchical event descriptor environment, because you have to take it beyond just a description of the EEG data itself. You have to annotate it and identify biological events, clinically relevant events in the system. And this has to be standardized across all of the EEG data across all of the countries. So the development of a hierarchical event descriptor is very important in the interpretation of EEG data. Coming to the crux of this meeting as relevant to our work in the Canadian context is HIBALL. And it's always a pleasure to meet my colleagues in HIBALL from the ULISH group with Katrin and her colleagues. I've been working with Katrin for approximately 20 years now, initially within the context of the International Consortium for Brain Mapping. And then we started to work on the big brain about 15 years ago. And then we finally evolved this into the Helmholtz International Big Brain Analytics and Learning Laboratory, which is funded on the European side by the Helmholtz Association, and on the Canadian side by the Healthy Brains, Healthy Lives network that I've just described. So I'll just give you a few examples of where HIBALL is today. You should come tomorrow. There'll be a lot more detail on where HIBALL is today, where it's going, but I'll just give you a flavor of it today. So HIBALL, its genesis is the big brain dataset, which many of you will have seen many times. But it's more than just an atlas at this point, just a template of a very high resolution 3D brain image. More and more we have examples of segmentations of specific areas, and I'm giving you an example of particularly relevance to many of us, the hippocampus. And as you can see here, this is a three-dimensional segmentation of the hippocampal subfields. And on the right, you see that represented in 3 dimensions and shown in the Atelier 3D visualization tool that was developed in Montreal, showing something that, in my ignorance, I was not aware of until I saw this, that the hippocampus has deep sulcal folds of its own. And as you can see in that bottom right view, very, very strong sulcation. I'll come on and mention that a little bit further in a moment. So HIBALL was developed to develop new simulation models using AI and high-performance computing, to use those tools to develop a transatlantic and interoperable platform for data exchange and collaboration, linking the European and North American initiatives, and to engage the international communities in imaging, AI, and brain simulation to advance neuroscience. The next 2 slides are somewhat out of date, and I think it's a testament to the power of the collaboration between the ULISH team and our team in Montreal. We started out with a discussion of the expertise in both groups. In ULISH, whole brain histology and cytoarchitecture, 3D architecture of axons, deep learning and machine learning, brain mapping, HPC infrastructure connected to Europe. In Montreal, deep learning and machine learning, 3D visualization with Atelier 3D, high throughput neuroimaging analytics, C-brain, human brain atlas and templates, big data compute infrastructure related to Canada. A lot of overlap, but also a lot of complementarity between the 2 groups. And as we've gone on in the last few years, there's been a greater degree of cross-fertilization between the 2 groups. And now at this point, you see both groups have projects working in both directions with a lot of complementarity between the groups. HIBALL is split into 6 work packages. I'll describe the work packages in a moment, but the most important part of this is at the bottom. Paul-Joanne Toussaint and Suzanne Wenzel, who are both in the audience, and this project wouldn't be proceeding without the management structure of these 2 individuals. This is a 3-ring circus, which requires a tremendous amount of coordination, and I think PJ and Suzanne are doing a superb job of pulling all of these groups together. We have 6 work packages on brain proscillation using AI-based segmentation, multimodal data integration into big brain, cellular segmentation. You've heard about this from, for instance, Katrin and Timo. Modular computing architecture for cellular big brain. That's where we're pulling together the IT infrastructures across the 2 continents. The virtual big brain is work that is an extension of work that has been championed by Victor Jirza and Randy McIntosh and Petra Ritter with the virtual brain. We're now incorporating layer information, chemoarchitectural information, and electrophysiological information to what will eventually become the virtual big brain. And lastly, the brain-inspired artificial intelligence, where we're developing machine learning strategies to allow us to better understand brain signal architecture. The joint infrastructure between the 2 sites that brings together C-brain and data lab within the context of, for instance, CONP or NeuroHub on the Canadian side, with the Helmholtz infrastructure at ULISH. In due course, this will, we hope, propagate into e-brains that you've heard about from Jan this morning. The idea ultimately is that you will have data that is resident in ULISH, be processed on the C-brain analytics in Canada or vice versa, so that the data is processed in situ. We can obviate the need to ship the data to the compute. We can now ship the compute to the data, which I think is a crucial step forward in global neuroinformatics. So far, HIBALL has produced 37 publications since 2020, many in top journals. And I'll just give you a few examples of the kinds of work that has been going on. It was very, very satisfying to see Jeff Mangin refer to Conrad Wagstill's work, where he showed that the depth of layer 4 in particular, but the organization of the cortex respects the equivolumetric principles of Bock in 1929, and showed that very nicely in this graph, that the depth of layer 4 is a function of the mean curvature of the cortex. And that's one example of this. Conrad developed an automated approach using machine learning to allow us to identify the 6 cortical layers of the human brain in both hemispheres using a fully automated approach. And many of you are very familiar with the Brodmann Atlas from 100 years ago. That was done with a very much a manual, labor-intensive approach. This is a completely automated approach using machine learning strategies, which takes us one layer beyond what Brodmann was able to achieve back 100 years ago. That was one example. There are many others that I could show describing the work of students engaged in HIBALL. Hannah Spitzer, Jordan de Cracker, Conrad Wagstill, Thomas Funk, Christian Schiffer, Edith Upschulte, are all students who've trained through the HIBALL program, and I think that these people are very much an example of what HIBALL brings to the table in terms of training our young scientists. Just more recently, we've started the integration of 1-micron sections. This is from the Jülich side, taking the data with the original big brain, which was 20-micron isotropic data. We're now moving it to 1-micron in-plane resolution, which allows us to start asking questions about the cellular organization of the brain. I mentioned the 3-dimensional chemoarchitectonic work. Thomas Funk did his PhD in my lab. He then moved on to work with Nicola, working on the 3-dimensional reconstruction of the original 2D radioimmunoassay data from Carl Zillis, Katrin Amons, Nicola Palomero-Gallagher, to develop 3D maps of the chemoarchitecture. So we want to incorporate that into the virtual big brain that I mentioned. You've heard from Katrin about the PLI, so I won't dwell on this except to say, as always, Jülich produces some beautiful videos of the organization of the hippocampus. It pains me to move us along, but you've seen these kinds of things already, so I'm going to move on. I mentioned the hippocampal work of Jordan de Cracker and the sulcation. Where we're going next is to take this kind of information and try and use the segmentation of the big brain hippocampus and its subfields as a template to segment individual hippocampi derived from MRI information. So this is a gold standard which can be used to understand the segmentation of the individual hippocampi from MRI, which is much poorer resolution, but you can use the big brain segmentation as a guide in segmenting that, and we can start to explore hippocampal changes in subfield volume, hippocampal sulcation, and explore the relationship between that and behavior, which I think is so far an almost virgin territory of undiscovered individual variability. All of this takes advantage, as you can see on that video on the left, of a flat mapping approach similar to what you've seen many times in terms of the cerebral cortex. You can apply the same cortical flattening approach to the hippocampal surface to allow a space for mapping across individuals. So in the last few slides, HIBALL is, apart from the science, it's producing a wealth of tools and data to make for the international community. You've heard about Zebra, but there are many other tools: display for 3-dimensional visualization, big brain warp, brain box, brain browser, Atelier 3D, microdraw, These are a variety of tools, some of which you'll be able to see in the demos this afternoon. Also the data you've heard about, the original big brain data set itself, the cortical layer maps that I've mentioned, and the cytoarchitectonic maps which Timo described and Katrin has described. Lastly, this is all just a teaser for the next 2 days. We have the 6th Big Brain HIBALL workshop today and tomorrow, the next 2 days, in Zadar. We will have international speakers. Last year we had Martin van den Heuvel, who talked to us about connectomics. This year we have Muming Pu, who's going to talk to us about transcriptomics, the next frontier, I believe, in understanding of brain organization, where we will still look at gene expression at an individual level. And I think incorporating that molecular perspective into the systems level connectivity perspective, I think, is going to open a whole new frontier of our understanding of brain organization. And we also have, I just mentioned the talk from Muming on transcriptomics. And we also have a HIBALL lecture in brain analytics and learning from Ivica Kostovic. So the world has come back full circle. We're back here again, and Ivica is going to talk to us about transient cellular compartments to the brain's network architecture, and we're all very much looking forward to that. So with that, thank you very much.